Welcome back to Biotechnology. So today we're going to think about errors in DNA or RNA copying, translation and repair of such things. So if we imagine our DNA is being copied, so let's have a separate strand, so open up a crack here and we will copy on a new section. Some things can happen. So obviously this could be DNA replication, in which case we're just making the other strand here and here as before, and it's writing in this direction, and we make an error. So let's have an error here at this point here. So in focus just about. So now we've got a problem with our new strand of DNA and the old strand of DNA is good. Then we generate a completely separate new DNA. So let's draw that a little bit down here. I think there's enough space. So we've got our new DNA and it's a complete strand maybe, but there is one base that doesn't match, which is about here. So it doesn't stick. It's got a little bulge and then it connects back up again. Possibly the next two would struggle to stick. Depends how it is. So now we've got a position that could theoretically be measured by chemistry as a problem, but how do we know which one is blue and which one is black? That is the problem for biology. So we've got this new strand, and if we copy it again, we're obviously going to get one that's wrong and one that is probably right. This is a problem that biology has sort of solved, in that it performs a process on the DNA after making it, which is called methylation. So the bases gain a chemical group on the side, a methyl group on the side, like this, some of them, not all of them, and that determines, it doesn't determine, it allows the cell to know, the chemistry of the cell to know which one is which. Because whether we've finished our DNA replication or whether we've got a partial strand, a molecule which is only this big is not going to be able to see the end to know which one is which, or even thread through from the end to get to here. It's a long way. It could be a long way. It could be millions of base pairs away. So it is difficult chemistry-wise, and if we imagine a copper cable where one of the strands is broken, it is also, without measuring resistance, it's also not trivial to work out which one it is if we're not at the break. If we're at the break, it is not trivial to work out which one of those strands it is on the other end if we want to pull it out. So we would like to maybe clip it and pull the whole thing out, but we can't do that unless it's color-coded in some way. And this is the equivalent, the chemical equivalent of a color coding. So now we've got all of our cables in our wire different colors, and if we know that the blue one isn't working, we can choose to not use that one or to replace it. So in this case, there is a mechanism to cut out this base and allow it to be replaced just in the same way as we bonded the two sections of this open section and another section with a ligase enzyme which connects back in another base and allows it to seal and connect back up again. So we've got perfect DNA again. So we can repair a single base error in DNA most of the time. Obviously, if the strand that we're trying to copy here is RNA then, that we're going to use to make a protein or something else, it is also possible, and it's easier to mark the RNA strand, but it is less problematic. So the consequences of making an error in an RNA strand is not as bad as the consequences of making an error in a DNA strand, because this is going to be a molecular machine, or it's going to go and make some protein. If we make some protein that doesn't work, for example, that is bad, but it's not super bad, as opposed to changing the code that was in the DNA so that the next cell is doing something different. So what we can see is statistically the chances of an error happening and staying in our RNA is 100 to 10,000 times as more likely as when copying DNA. So we've got our error in the RNA, and there are several things that the cell can do to get around this. So one of them is the specific alterations, the specific errors that are more common are just some of them. So some, I think it's C to U alteration is quite common, other alterations are less common, and therefore, because we have a group of three, so we have we have 64, I believe, combinations, and we only have 20 amino acids plus a start and a stop codon and maybe some other codes, but we've got effectively three-ish codes for each amino acid that we could use. We can include a little bit of variation that we'll get the same amino acid even if we end up with a common mutation. So that can make this process more stable to accidental mutations effectively, although it isn't always the case because it's biology. It doesn't have to be the most efficient answer. There are several other types of mutation that we could have. We could have an added base or a deleted base, so we don't just have to change one base. We could change the number. That is more significant depending on where it is. So if we have, for example, the start codon of our RNA, we're making some RNA here, and this RNA has an extra base here, the first amino acid will be correct, but the second amino acid will be shifted by one, so it should really be these three, and it's actually this one, it's actually 1, 2, 3, 1, 2, 3, so it should be those three, but it's these two and the extra one, and then all of the rest of the amino acids in our whole protein will be wrong because they'll all be shifted by one letter backwards. This is known as a frame shift mutation, so effectively the start codon, the beginning of the protein, defines where we want to start our protein, and then we continue from there, which sets our sets of three up, and if we add or remove a number of bases that doesn't divide by 3, then we end up in a different frame, so we end up reading the wrong bases together, which means that we get completely the wrong thing. It's not just a little bit different. None of the amino acids have necessarily anything to do with the original ones, and if they do, then it's just by chance. So we end up with a big problem. Or something completely different. Typically that will inactivate the protein completely. If it's just an RNA copy, then obviously this piece of messenger RNA is not going to do anything useful, but that doesn't make it completely ruined. If it's a DNA error, which, as we mentioned, are rarer, it's obviously far more serious because we then can't make any mRNA that works. Last time we were talking about PCR, which is making sections of DNA and copying them. So we've copied a whole load of DNA, and it's double-stranded DNA because we've allowed it to form 2 strands. We haven't heated it up for the last step. If we extract that from our crime scene, the question is, and how to get it back to the same theme, is how do we tell the difference between the DNA of person A and person B? Well, it's difficult. It's difficult because it isn't obvious where any information is that we might want to use. So if we know that the criminal, if we know some information about the criminal, maybe they're white or maybe they're black, that doesn't help us massively because the information of what color skin they have is not very easy to extract from the DNA, not very easy to impossible. We can have two people who look completely different but have almost identical DNA, and the other way round. We can have people with completely different DNA that happen to look very similar, and so it's not a brilliant method to use. It's fairly difficult to even tell very general things. The best we can do is gender. And that will be physical gender, an original physical gender, and not necessarily what the person looks like either. And so that's also not 100% useful, depending on what information we have about the person who we imagine has done this crime. So those aren't necessarily useful pieces of information, and from a scientific point of view, not a great way to start, because we're already biasing our study. So what we really want to know is, can we get any information about the difference between people from this DNA? And so we would maybe look at a gene for a protein, for example. But that is super ineffective, because if we've got a gene for a protein, maybe actin. So actin is one of the proteins in muscles. That's why I put my hand in the screen and moving it. It's kind of a dumb way to do it, sorry about that, but that was the only thing that occurred to me at the time. So actin is one of the proteins in muscle, one of the two main proteins in muscle. And unfortunately, for the purposes of identifying somebody, if we have a mutation in actin, so we've added maybe got a frameshift mutation late on in the protein, so it's not completely damaged, or we've got a few alteration mutations, so we've got different amino acids at different parts. It doesn't take very much for the actin to not work, and that would mean that the person would be inviable, they wouldn't live. What we notice from that is that actin has an incredibly high equivalence between people, but also between people and animals, even down to bacteria. So all cells have an actin skeleton, which they use to move things around the cell. It's exactly the same chemical mechanism that moves things inside the cell as it does to move your arm. It's exactly the same chemical process that's going on. It's a fiber that's getting shorter. And that is important because it means that there are very, very few variations in actin that can actually work. And although they're known and we know which organisms they have them, we cannot tell the difference between two people from differences in actin. They have the same. If we look at, for example, skin color, so we would look at the genes for making melanin. Most of those are the same too, because they're either active or they're not. So in white people, some of them are deactivated so that they don't have as much skin coloring. That doesn't mean that they don't have the genes for them. There is a possibility that they can then mutate, obviously, but that's only tiny alterations. And as we mentioned, it would only take a frameshift mutation, the addition or deletion of a single base, to completely inactivate an enzyme or a protein. And so it doesn't take very much, and we may not find that. We'd have to read every single letter in the code to be able to find that, and it may just be one. There could be several. Is there a difference between the two? Well, if it doesn't work, practically no. They would look identical, the people. But physically, yes, there's a difference. So there will be something that we can't tell, and the probability of those happening is not great. So what we want is really something that is simple and that is very variable between people so that we can make a test. So if we look into it a little bit further, we would see that maybe we could watch a crime drama or something, and we would see that a DNA test would look somewhat like this. You end up with some stripes, and they mean something, and the people in the shows tend to pretend that it means a lot more than it really does. But what we actually end up with is some stripes, and that should remind us of something. It should remind us of chromatography, because if we take a piece of paper and we put a dot on it of ink, for example, this blue, and we put it into a solvent that is not great for the ink, but is good enough to get it to dissolve a little bit, it will move up as the solvent moves up the paper, and we will end up, I shouldn't have drawn lines, let's draw it without lines, we'll end up with a dot of green. If it's blue, we'll end up with a dot of green and some red, maybe a little bit of black, depending on what dyes they've used in the ink. That is exactly the same process as we want to use here, but we would like to use something that divides our DNA by some characteristics that are related to differences between people. Luckily for us, there's a lot of DNA that doesn't code for proteins, and that is far more variable than the DNA that codes for proteins, because you're not going to die if that's a bit longer or shorter. That piece of information is typically a repeating sequence between genes, and it doesn't matter how long or short that is. It tends to copy itself, so it's got its own little genetic advantages to being copied. So it tends to copy itself. It could be a fossil of being attacked by viruses because they add their genetic material and then it gets deleted again. So it could be, or in some cases it is, a fossil of being attacked by viruses. And then the length of that varies between people, and it's closer, and they're in the same place, depending by relatedness. So it's more similar to people who are closer related to each other, and more different to people who are more distantly related to each other, which means that we can also use it for a paternity type of test. So what we're interested in is a section of DNA, and it doesn't matter which section, but obviously more is better. And we want to cut it up, and we need to cut it up always at the same place. So we don't want to cut it up in the repeating section, because that will be bad. Then we'll get lots and lots of really tiny bits that are all the same. That's unhelpful. We would like to cut it up inside the, so this is maybe our gene, and then we've got a repeating section afterwards, and we would like to cut it inside the gene. So let's have another gene down here and cut it inside the gene. And then we've got a section of repeating units that don't code for anything in between these two genes that is of variable length, and that is what's going to tell us whether it's the correct person or a different person. And as we see, it obviously has no information whatsoever about what the person might look like or be like or anything, because this is the non-coding section. There is research that shows that it isn't entirely non-functional. It does some things. It influences how effective the neighboring sections are. But for the purposes of this, we're using it because it's not so functional, because changing it doesn't stop the person from living. And so that means that this is much more variable than this section, which is our gene for whatever it is, some protein, because if we change that too much, it will stop working. So if we have a mutation here, an error in here, it stops working, so we can't use that piece because that's almost always the same. So while we're on here, let's have a practical look at how we get to this particular diagram, particular result. This is not a diagram, it's a result. It will be a measurement that looked like this. How are we going to get there? We need a gel. It's called gel permeation chromatography. No, it's not. It's called gel electrophoresis. Sorry, that's exactly the wrong answer. Gel permeation chromatography is the other thing. It's easy to get confused between them, and I obviously just did. So this is gel electrophoresis, and the important thing about gel electrophoresis is that the speed that something moves is dependent on its size and size to charge ratio. So what we've got is we've got a string of DNA, and the string of DNA has phosphate groups on it, so it's got negative charges all along, approximately 1 per base. This string of DNA, or we could do it with RNA, but we're doing it with DNA at the moment because of PCR. It's a lot easier to get lots of the same DNA. It's hard to get lots of RNA that's all the same. We'd have to make DNA and then make RNA out of it. So we've got our single-stranded DNA or double-stranded DNA Now let's have a double strand. It's got negative charges all along. I've drawn negatives on it again because I made that the ladder. So it's got negative charges on it all along, and because its charge per unit length is constant, it moves fairly constantly. But if we slow it down with something, so we need a medium that is tortuous, we need some kind of particles in the way that it has to get round, we need a gel, then this DNA has to sort of force its way through the gel, and so the long pieces will stay down near the bottom and the short pieces will move faster. That would be what these are. So we apply some kind of electric field, so we'd have to apply a positive to the top and a negative to the bottom, generating an electric field that would drive the movement of these DNA molecules up, in this case, up the gel. Usually it's done flat, so that we don't have to worry about gravity. So we're driving it up the gel, and the resistance to it is physical. It's got to get through a gel. It's got to get between the particles, it's got to get between the network of the gel. Typically this would be a polymer gel. What I've done here is a sample, so this is my sample, and next to it is a reference, and that's because it's quite difficult. It's quite difficult to get a standard because we've got a voltage, it's typically 20 volts on here. It's going to change the temperature. The rate of movement is dependent on the DNA and what its charge ratio is, but also exactly how the gel was made, which is quite tricky to get it reproducible, and also the temperature. And the problem with the temperature is obviously because we've got 20 volts across here and we're running a reasonable current, it's going to change temperature during measurement, in most places at least. And so we're going to end up with a problem with temperature, with fixing the conditions. It's a lot easier to run a standard. So this would be a standard, you can buy them. It would be the DNA from some kind of bacterium typically, because it's easy to make a lot of it. And then we know where these peaks should all be, and so we can use those as a reference. If we run them at exactly the same time, we can use them as a reference for our measurement bands, so that when we come tomorrow and do whatever suspect number 2, we can then shrink and compress the two scales to make them fit to each other without having to do them on the same day. Obviously, ideally, you do them on the same piece of gel in the same run, but it's practically better to run an internal standard so that we know exactly what the number is, what the length of this one is, because we can count up from one end down or from the bottom end up. This is the short one, this is the longest one. We can count from one to the other. I accidentally mentioned GPC, which I didn't mean to, but I've done it now, so I guess I'll have to explain what it is. Some of you are from biomaterials, many of you are from biomaterials, many of you will know what it is, but I will try to make you aware that this is a problem. So it's not a problem, it's an easy thing to get confused with. That was why I got confused. GPC is where we have also a polymer or some kind of molecule, and it's trying to get through beads. So we've got some beads, and there are two possibilities of this, whatever it is, molecule. It can either go into the bead, which has got pores in it, like that with dots on. It can either go into the bead or it can go past. The problem, the sort of mental problem that this generates is if the polymer goes into the bead, the shorter ones will be slower, because the shorter ones will preferentially go into the holes in the bead and get stuck. And so they will be stopped, and the rest of it will pass between them and go faster. In this case, we've got no pores in our beads, if we consider it as beads. If we consider it as a network, we can say it's going through the pores. It depends how we want to view the gel. But in this case, we have the moving phase, which is the because we're driving it electrically, it's just the DNA. The moving phase is the DNA, and it is not getting stuck inside the beads, it's instead just getting blocked by them. In this case, we've got the stationary phase, which is the bead, and the polymer is going inside it, and so preferentially smaller polymers go inside it, and so we will actually get the opposite type of extraction. The shorter ones will end up at the bottom, and the longer ones will go straight through because they can't go inside. Now we have a new problem that we've generated here because we've got a positive electrode, so OH minus is perhaps going to get discharged. If we take the charge off OH minus, we're going to have to take two of them to do that. We're going to be removing OH minus ions, or we could take H2O, and we can remove an electron from it, generating an H plus, attach it to here so that we can get all finished. Effectively what we're doing is we're making H plus here. We're destroying OH minus, so this end is going to get acidic, and this end is the opposite, so it's going to be dumping electrons into the solution, so we can add negative electron to H plus, making it into H2. We're going to end up with oxygen from here, and the oxygen is effectively leaving the H plus over, and in this case we're making hydrogen from here, and that's going to be removing H plus, and so we're going to end up with OH minus at this end and H plus at this end. We're going to end up with a pH gradient. That is sucky because we don't want that. We don't want to generate a lot of negative charge down here. We don't want to break down our DNA in this case, or our gel. And so typically this is run in a buffer, which is carefully chosen. The strength of the buffer is carefully chosen to be able to handle what kind of conditions that we want. Because obviously we don't want very strong ionic strength for a reason. Why don't we want very high ionic strength? If we put a lot of buffer in there, we could have a very high ionic strength. We could put in citric acid, for example. On this OH end, we can add a lot of OH and nothing is going to happen because it's just going to react with the citric acid. We could do that, but it doesn't work very well because if we have a lot of ions in here, we've got a charge distribution. The ions are going to be the thing that move, and our large heavy DNA is not going to move as much. It will be shielded from the charge distribution. So if we put in, let's just put in sodium and chloride. So we put in sodium plus and Cl minus into here. The sodium plus would all come down here. Let's rub out these temporarily. So we'd end up with a lot of sodium plus at this end, and we would end up with a lot of chloride minus at this end, and our effective charge would be reduced, which you know from physics as exactly what happens in a capacitor. If we take a capacitor, the dielectric does that. We end up with a charge close to the positive electrode and a charge close to the negative electrode that opposes it, and it means that the two electrodes don't see each other as much, and so we can get more charge on it as a capacitor, or we know it from other courses that we might have had. Depending on which course you're from, we should be familiar with this effect that close to an electrode, ions of the opposite flavor, ions of the opposite charge will gather up, and that will mean that the field inside the solution is much lower than the field outside. That's why we have to add such a stupidly high voltage, because most of the charge is wasted in moving these ions around, and we don't really need to do that. That's a waste of money. It's not super expensive because we're going to get a lot of money doing this test, but it's very inefficient because most of the thing that we're doing is heating up the environment, making acid, moving these sodium ions and these chloride ions around against a concentration gradient. So we're doing something unhelpful by accident. What we really want to do is move our DNA. So ideally we would have as low a concentration of other ions in there as possible, but we are fighting the other problem that if we don't have any buffer in there, the pH will go crazy at the ends, which we don't want either. So after that long aside, back to the main theme, which is mutations, effectively, errors in copying DNA and RNA. And if we have an error in copying our DNA, most of the time, nothing happens. It will very often be in between the regions. It will very often be in one of those regions that don't do so much, and so nobody dies. In a cell, a human cell, we have 2 copies of the DNA for most of our proteins. Some of us have only 1 copy because one of our parents had a damaged or gave us a damaged version. Most of us have 1 functioning copy of some things and 2 copies of most things. If we then make RNA from that, and then half of it isn't doing anything useful because it's a copy of a non-functioning gene, but that doesn't necessarily hinder us. Where it becomes critical is if it's one of the genes that modify cell growth, because that will make it into a precancerous cell. So topical would be the virus that gives us, or some of us, cervical cancer, for example. It is supposed to be responsible for other cancers in other people, so it's not just a sexist cancer. And what it does is it infects our cells and inserts its genetic material somewhere in our genome. Most of the time that's not a problem, because most of the time it doesn't hit something that is important. Some of the time it hits an important functioning part of the cell and the cell dies. So if it's one of the copies that we cannot replace because the other one doesn't work anyway, the cell might just die. If it makes the cell do something that kills it, it will also die. So it makes it make something that's poisonous, or if it makes it kill itself, then it will just die. That is not such a big problem to us. What is a problem is if it interferes with cell growth or interferes with the process by which cells kill themselves when anything is going wrong. Then it becomes a precancerous cell and we have a problem. So now I'm interested in what happens then. So we've got some kind of mutation in our DNA and it's not correct. It's not the one that we want. So we've got some kind of problem here. Oh, that doesn't look very good. We've got some kind of problem in our DNA and we've generated a new strand and it's wrong. But we've left it for so long that it's now difficult to tell which was the original strand and which is the old strand. Which is the new one. So we don't necessarily know which one it is anymore, but we can still fix it. What we need is some kind of template of correct information from somewhere. So we need a strand of DNA or RNA that contains the correct information, and we want to copy that into this gap. There are several equivalent mechanisms for doing this, which we will introduce gradually. What we will effectively do is assign this piece of DNA or RNA to the strand that we want to repair, and then we need to open it up, cut it out, and then copy the information of this repair strand into it. That would be a way of fixing these errors. It's difficult and doesn't always work, so we still get cancer. One of the known human cases where this happens is the BRCA gene, which is the one that is mutated to the BRCA1 version in people that have a tendency to get breast cancer. If it's in the BRCA1 version, it doesn't work very well, and so they can't do the thing that they're supposed to do. And what it's really doing is copying in a section of DNA and fixing a problem. There are a set of different problems that could have occurred to generate this, but the one that the BRCA is particularly specialized to is a double-strand break. So what is happening is some radiation comes along or some bad chemicals come along or an accident happens during copying and we end up with a broken strand. The problem with having a broken strand is there is no information on the end of the strand to say that it belongs to here. And there is no information on the end of the strand to tell us whether there's a missing nucleotide in between, which I just now rubbed out, I think there were 2, whether there's a missing nucleotide or 2 in between, or whether there's an extra one on the end. We don't have that information. If we have a straight break of our DNA, it's a big problem. It's very difficult for the cell to recognize that and stick it back together. And one of the solutions is to take a template piece of DNA and use it to check. Effectively, we will take this strand, which is the other side, bring it in here and copy it to make it match this single strand. Then we can bring it back over here and we can copy the other side in. And that will mean that we can fill in the gaps, and it will also mean that we can align the 2 ends, because this can be quite long. So we have a way of fixing a break in DNA that is very, very bad. This is also a mechanism that we can abuse. Because we are biotechnologists, we could change this sequence here and use it to change the DNA. We would obviously have to initiate some kind of break here, and there are better ways of doing this using a related but not quite the same mechanism.